Here we are going to learn about what is protein and classification of protein. So this is the classification of protein. Proteins: simple, conjugated, and derived. There are three types of protein: simple, conjugated, and derived. Globular protein: simple are again divided into two parts: globular protein and is scleroproteins. The first one is albumin, globulin, gluteins, prolamines, histones, globins, protamines, and lectins. Scleroproteins: collagens, elastins, keratin. Conjugated proteins are the nucleoproteins, glycoproteins, mucoproteins, lipoproteins, phosphoproteins, and chromoproteins, metalloproteins. Derived proteins are primary coagulated proteins, proteins, metroproteins, proteases, peptones, polypeptides, and peptides. Simple proteins. Here are spherical or oval in shape. These are spherical or oval in shape, soluble in water or other solvent, and digestible. Albumin, soluble in water and dilute salt solution, and coagulated by heat. Serum albumin, oval albumin, ova albumin, lactalbumin, globulin soluble in natural and dilute salt solution, serum globulin, betalain, bulletins, soluble in dilute acid and alkaline, and mostly found in plants, lutein, originin. Prolamines, soluble in 70% alcohol, gliadin, wheat, zein, maize, stones, strongly basic protein, soluble in water and dilute acid, but insoluble in dilute ammonium hydroxide, thymocysteine, globins. These are generally considered along with the histones. However, globins are not basic proteins and are not precipitated by NaOH. Protamines, they are strongly basic and resemble histones, but smaller in size and soluble in NH4OH. Protamines are also found in association with nucleic acid. Examples: sperm proteins. Lectins are carbohydrates binding proteins and are involved in the interaction between cell and proteins. They help to maintain tissue and organ structure in a laboratory. Lectin are useful for the purification of carbohydrate by affinity chromatography. Example: concanvaline and A-glutine. Fibrous proteins. These are the fiber-like shape insoluble in water and resistant to digestion. Albuminoids, scleroproteins are predominant group of fibrous proteins. Collagens are connective tissue proteins lacking tryptophan. Collagens on boiling with water or dilute acid yield gelatin, which is soluble and digestible. Elastins. These proteins are found in elastic tissue such as tendons and arteries. Keratin. These are present in exoskeleton structure, hair, nail, horns, human hair. Keratin contains much as 14% cysteine. Conjugated protein. Nucleoproteins, nucleic acid, DNA and RNA is the prosthetic group, e.g. nucleohistones, nucleoprotamines, glycoproteins. The prosthetic group is carbohydrate, which is less than 4% of protein. The term mucoprotein is used if the carbohydrate content is more than 4%. Lipoproteins, protein found in combination with lipid as the prosthetic group serum lipoproteins. Phosphoprotein, phosphoric acid is the prosthetic group, is casein vetillin. Chromoproteins, the prosthetic group colored in nature. Example, hemoglobin, cytochromes. Metalloproteins, these are proteins containing metal ions such as Fe, copper, cobalt, zinc, manganese. Example, ceruloplasmin, carbonic anhydrase, derived protein. The proteins are of two types. The primary derived are the denatured and coagulated or first hydrolyzed products of protein. The secondary derived are the degraded due to breakdown of peptide bonds product of proteins. Primary derived proteins. Coagulated proteins, these are denatured proteins produced by agents such as heat, acid, alkali, cooked protein or coagulated albumin. Proteins, these are the earliest product of protein hydrolysis by enzyme, dilute acidic acids, alkalis, etc., which are insoluble in water. Fibrin formed from fibrinogen. Metaproteins. These are the second stage product of protein hydrolysis obtained by treatment with slightly stronger acid and alkali metaprotein. Secondary derived protein. These are the progressive hydrolytic products of protein hydrolysis. These include proteases, peptones, polypeptides, and peptides. Nutritional classification of proteins. The nutritive value of proteins is determined by the composition of essential amino acids. From the nutritional point of view, proteins are classified into three categories. The first one is the complete proteins. These proteins have all the 10 essential amino acids in the required proportion by human body. No promote good growth. Partially incomplete protein. The proteins partially lack or more essential amino acids and can promote moderate growth. Wheat and dried protein. Incomplete proteins. These proteins completely lack one or more essential amino acids. Hence, they do not promote growth at all. Gelatin. Biologically important peptide. Several peptide occurs in the living organism that display a wide spectrum of biological function. Generally, the term peptide is applied when the number of constituents amino acid is less than 10. More examples, some examples of biologically active peptides and their function are described here. Glutathiones. It is a tripeptide composed of three amino acids. Chemically, glutathiones is gamma glutamyl cysteine glycine. It is widely distributed in nature that exists in reduced or oxidized state. 2GSH GSSG functions. It is steady state. In a steady state, the cell generally maintain a ratio about 100 upon 1 GSH GSSSG. The reversible oxidation reduction of glutathione is important for many of its biological functions. Glutathione serves as a coenzyme for certain enzymes, prostaglandin PGE2, synthetase, glyoxylase. It prevents the oxidation of sulfhydryl groups of several proteins to disulfide groups. This is essential for the protein function include that of enzyme. It is believed that glutathione's association with glutathione reductase participates in the formation of correct disulfide bonds in several proteins. Glutathiones is involved in the transport of amino acid in the intestine and kidney tubules. Gamma glutamyl acid, glutamyl cycle, Mr. cycle. Glutathiones is involved in the deoxycitation detoxification process. The toxic substance organophosphate nitrocompounds are converted in meracarboxylic acid. Toxic amounts of peroxidase and free radicals produced in the cell event. Glutathione's peroxidase is a selenium containing enzyme. Tryptophan, thyrotropin releasing hormone. It is a tripeptide secreted by hypothalamus. TRH stimulates pituitary glands to release thyrotropic hormone. Oxytocin, it is a hormone secreted by posterior pituitary glands and contain 9 amino acids. Non apeptide. Oxytocin causes contraction of uterus. Vasopressin, antidiuretic hormone, is also non-peptide produced by posterior pituitary gland. It stimulates kidney to retain water and thus increase the blood pressure. Angiotensin, first is a decapeptide with which is converted to angiotensin. Second, 8 amino acid. The latter has more hypertensive effect. Angiotensin second also stimulates the release of aldosterone for adrenal gland. Collagen is the most abundant protein in mammal. It is rich in hydroxyprotein and hydroxyl cysteine. Several biologically important peptides. Methionine L-encaphalide. It is a pentapeptide found in the brain and has opiate-like function like inhibit the sense of pain. Bradykinin and calidin. They are nona and decapeptide respectively. Both of them act as powerful vasodilator. They are produced from plasma protein by snake venom enzyme. Peptides, antibiotics such as gramicidin, bacticidin, triacidin, and actinomycin are peptide in nature. Aspartame, dipeptide, aspartic acid, and peripheral. These are some multiple choice questions. The amino acid found in protein structure. The following is non-protein amino acid. The bond in protein structure that are not broken or denaturation. Sequestrator, automatic